Good afternoon. My name is Morgan Levine. I'm from the Department of Human Genetics at UCLA. And today I'm going to discuss what I'm going to call a potential biomarker of aging. I'm not sold on this yet, so you guys can be the judge, which we are calling the epigenetic clock, and this is based on DNA methylation levels. So traditionally, we've used the mortality rate to kind of measure the rate of aging in different populations. However, this is kind of an archaic notion. So we use what is called the Gompertz law of mortality. This seems to be consistent across, at least in humans, a number of populations. It basically shows that the rate of death increases exponentially with age, following about age 30, so after growth kind of ceases. However, we typically don't want to have to wait, at least when we're studying humans, given their lifespan, till a person dies to figure out whether they were aging faster or slower than we'd expect. And I think this has kind of come up a lot today, is we'd like a biomarker where we can actually monitor the rate of aging at any point in the human lifespan. And this will allow us to test whether interventions work, to test the mechanisms of aging to try and figure out what causes certain people to age faster or slower. And also it can help with early detection to figure out who's most at risk of different chronic conditions or death. So this is where we are discussing what we're calling biological age clocks, also could be biomarkers of aging. And this is really a method to estimate the age in years of a biological sample. This can be a human, a cell, tissue. So a few examples of the types of data that people have used: telomere length, gene expression data, protein expression data, and DNA methylation levels. And we're hoping that with these, maybe we can do better than kind of these come to the fair, I'll guess your age by looking at you. So this is, I promise, my only slide on telomeres. I'm not going to talk about telomeres. This is kind of the most popularized way that people have thought that we can measure aging, because telomere length seems to correlate fairly well with chronological age. At least in this sample, it was correlated about 0.45. However, there's some controversy on whether this is actually a biomarker of aging. So then people started to actually use gene products, so expression levels, protein levels, and they were starting to get slightly better correlations with chronological age, about 0.63. However, Steve Horvath, who is my PI, actually used DNA methylation levels, so epigenetics, and he was able to get measures that correlated with chronological age at 0.96. And this was consistent across a number of different types of tissue. So I'm just going to briefly describe how we measure this. So he uses the Illumina DNA methylation array. This has 486,000 locations. And for each CpG site, it specifies the amount of methylation that is present, and it's continuous from 0 to 1. So to construct this, we use something called an elastic net. It's a penalized regression model. Basically, it allows you to consider more covariates than you have samples, because in a normal regression, you actually can't run it when you have more covariates. So our dependent variable was just chronological age in years, and the independent variable was the 486,000 DNA measures. So this data included 4,844 non-cancer samples from 82 individual data sets, and with 51 different types of tissues and cells. And it was largely healthy tissues. We specifically excluded cancer tissues. So the first 39 data sets were used to what we call train the data, so build our equation. And then the rest of them were used to predict or actually see if we could validate it. So the training data set was chosen to represent a wide spectrum of different tissues, so a diverse array of tissues, and also involve a high proportion of samples that had the 450K Illumina chip, because it can also be measured on the 27K. So we ended up excluding and only studying 21,369 CPGs because they were on both chips, and they had to have fewer than 10 missing data sets. So what the elastic net does is it actually will select the covariates that are most important. So it ended up selecting 353 CPGs, and basically it constructs your epigenetic age based on a weighted average by these with the regression coefficients across these 353 sites. So this is a really messy slide, and I don't expect you guys to read it, but basically it's showing the correlations with chronological age across different tissue sites and in different samples. This, however, I want to point out, is in the training data, so we'd expect it to be very highly correlated because this is, you can think it might be overfit. But you're getting correlations of around 0.97 with chronological age. Some of the lower ones are around 0.8. But then when we look at the test data, which is a completely independent sample, it had nothing to do with our building of the model, we get, again, really high correlations. So when we look at all the test data up here, it's hard to see, but we get a correlation with chronological age of 0.96. So it's doing a really good job of predicting how old someone is just by knowing their methylation pattern. We also find the same patterns. This again was in the test data, the validation sample, in brain tissue. So up here we're getting correlations 0.98. This is a little lower, 0.81, 0.99, so almost perfect. And then there's been a lot of talk about stem cells. We actually can look within stem cells and see that they actually are 0 years old, which is what you'd hope to find. And then we can compare them to other tissues and cells, which tend to be about the same chronological age as the donor. We also found that this is highly heritable. So in the twin sample, using twin studies, in newborns there seems to be about 100% heritability. This bottom is the newborns. And in the adults, this decreases to 40%. So this could be environmental perturbations, which are kind of changing the rate of aging. We also found that obesity will accelerate epigenetic age in liver, but it's not accelerating it in muscle, blood, or adipose tissue. So finally, what we're doing now is we're trying to see is this predicting some of the age-related conditions that we'd expect. So we're finding that even after adjusting for chronological age, even though they're very highly correlated, knowing your epigenetic age, so knowing basically the difference between your chronological age and your epigenetic age, is a risk factor for all-cause mortality. And it also seems to be associated with both physical and cognitive functioning. And we're doing a lot of other outcomes of aging, but they're very preliminary, so I didn't feel like I could show them here. So just in conclusion, epigenetic age acceleration could be a useful mechanism for studying, or a useful measure for studying the mechanisms and consequences of aging. The jury's still, I think a lot of work needs to still be done to whether we can decide that this is truly a biomarker of aging. That being said, if you want to study it in your lab, there's very user-friendly software for actually calculating this measure, assuming you have the 450K or 27K chips. And we're always happy to collaborate. And I put this actually in a few days ago, but it fits perfectly with kind of what you guys have been talking about. What I'm wondering is, do these systematic changes that we see with time, are these more evidence of the damage accumulation theory or the programmed rate of aging theory? I think there's actually evidence for either one. I don't think it points to one or the other. And then just acknowledge my collaborators.
I don't understand one thing that, well, maybe I don't understand many things, but when you look at the changes in methylation as a function of chronological age in various tissues, are you looking at the position of each of the methyl CpGs in the DNA and watching it change, or are you looking at the overall landscape of CpGs?
It's more the latter, the landscape, so kind of the profile that seems to correlate with age.
And there's no localization of the CpGs in the algorithm at all. It's all.
Well, there's specific sites.
Oh, there are sites.
Yeah, it is.
Okay, that's what I was really asking.
Oh, yeah, sorry.
So
there's 353 specific sites that
and these
vary
regulate 363 regions of chromosomes with genes in
they're
them.
different loci within the genome.
And so that information as to what sites change as a function of chronological age would be an interesting thing to explore in itself, right?
Yeah. We also don't think that there's we don't think there's a few drivers in there that are kind of driving the whole effect, because we have done some.
So if you remove some and then
we've done
look
some
at,
leave some out
right.
analysis, and it seems to predict, quite on par with what we're getting with all of them. So, yeah, I think it's.
And these functions don't fall into Go categories or things like that?
I believe that we have, I don't know the gene ontology categories off the top of my head, but we have looked at that. We have looked at different pathways.
Okay.
So.
Thanks.
One quick question and one maybe not so quick question. The quick question is, so do you do the fitting separately for each tissue and find different?
No, all the tissue are in together.
All the tissues are just run together. So there's one set of 353, whatever that fit for. Okay. The harder question is, do tumors age?
So I know Steve did have a paper. I'm not an expert on his paper. He had a paper on tumors that showed they did age. I don't remember the exact results, but I can find the citation for you.
I'm just wondering if you can look at a tumor.
Or no, I think they were epigenetically younger, not.
Well, but did they retain some memory of how old the cells were when the firmware was first created?
I'd have to find the citation. I don't know that off the top of my head.
I assume that all of the studies that you report were done with human tissue.
Yes.
Have you looked at other species?
So we're actually doing mice now. We don't have anything.
Any data.
Yeah, we haven't done it yet.
One thing you might think about is doing this with tissue from animals that allegedly do not age.
The negligible senescent phenotype.
Yes, and that tissue should be easily available.
Catch on fish.
I don't understand the technical side of this. So how did you select these 363 sites?
We didn't select them. The statistical model selected them.
Okay, so they were selected as a good predictors, right? Do you know their localization in genome?
Not off the top of my head, but I could get you the list.
But are they are they marking certain genes, or it's like some intergenic spaces, repeats, or something like that?
I think it's a mix. Yeah, they're not. Some of them are in.
And we all know that during aging, both in culture and there were lots of reports that generally there's a hypomethylation of DNA is happening with time. Do you see that the patterns of changes really indeed involve demethylation rather
Some of them do. So the majority are increases in methylation, but we do have, I don't know the exact number, but a significant proportion which are actually declines in methylation.
The last question, is there anything we can read about this which is published already?
Yes, so the first paper which talks about all this stuff is right here.
Thank you.
Yeah.
was just going to say that there's a big literature emerging in oncology where, for example, DNA methyltransferase mutation, which is inactivating, then leads to demethylation generally in DNA and a lot of CpG island hypermethylation. It's not totally understood why that happens, but I think it would be really interesting to figure out whether or not, and that's a particular phenotype and a particular set of CpG islands, whether that's a match for what you have.
Yeah. So this is done in all non-cancer, so I don't know if that would make...
Right, no, that you would now take your signature and
Yeah.
put it with cancer and see whether they end up being the same, because it sounds very similar.
Yeah, that's a good point.
Yeah, I want to ask, the one sentence summary, what I want to ask is what's going on. But let me make it a little more crisp. It's not news that a body changes as you age, okay? And a lot of stuff is going on. And one of the problems with these giant assays, okay, is that the computer is doing something, but you have no idea what it's doing. And I can't tell if there's any content in this result at all. Maybe it's just one of a million things that are happening as you age, and you just happen to find, one of the many, many, many signals. I find myself wondering, for example, I presume this is what's the singular value decomposition? What did the next best eigenvalue do? So this is the best correlated. What did the next best one do?
It's actually not. So it's it doesn't use eigenvalues at all. It's a form of regression. So it's, if you think of like an OLS.
The point is, it's an algorithm that you can't explain very carefully, and there's a second best solution. And there's a whole bunch of questions you can ask about what's the meaning of what it found that are kind of difficult when it's in a black box package. And the thing that's going through my head right now is that I don't see any evidence that it means anything.
I would say that this is less black box than, say, a random forest algorithm or a principal component algorithm.
I know that's your opinion, but you haven't given us any
I
reason
could
to think
show
that.
you the actual model that shows the weights of each of the different CpGs. And
I'm giving you a hard time on purpose,
Okay,
but the
that's
point is,
fine.
now come back to my original question.
Sure.
Tell us what's going on.
I don't know what's going on.
I thought so.
Yet.
I think it's interesting because this probably is linked to the biology of cancer and why we have more cancer as we get older, and the whole field concept. That when I showed the clonal evolution, that first hit is not even. They find the first hit to be the DNA methyltransferase mutation that's not even in people who have leukemia. So, and people are walking around already with aberrancies in their DNA methylations. I think these are linked. We just have to figure out what it means. I agree.
Yeah,
with that.
I agree. I'm not conceited enough to say I know what's going on at all. This is a brand new thing.
But it's created a permissive environment.
Yes, it does. I wasn't going to share any preliminary results, but we have found that it does predict cancer onset for certain types of cancer 15 years prior. So there is something going on. I also don't think it's the only thing. I agree, this is not the aging process by any means. I don't think it's the only thing going on. I think it could be useful for studying mechanisms along with probably dozens of other measures.
So I have a very simple, naive question.
Okay.
You keep mentioning the fact that there's a clock, right? A clock to me is that something that ticks every certain amount of time. So assume that if you want to use the methylation level as a clock as a marker for the age, that means you know the underlying dynamics of the methylation and demethylation will have a time scale, and that's your ruler of time. And that somehow, from the methylation pattern, you actually can infer how many such reaction has happened, therefore how old the person is. Do we know enough about the methylation dynamics and demethylation dynamics to know that there is a foundation? There is a foundation for this relation between the methylation pattern and the age?
I personally don't.
That's where I was going with my question.
Yeah.
So let me just make the analogy that I think everybody in the room would agree that as we age, there's a mutational accumulation, which has something to do with the cell division rate and something to do with the environment and a complex set of variables. And just as we make mistakes as we copy DNA, we probably make mistakes as we copy epigenetic marks. And those mistakes are not going to be uniform across the genome, just as the mutations are not uniform across the genome because the genome has sequence contexts that prefer certain mutations with certain carcinogens and things, or mutagens and things. So you may have picked up areas which are error-prone for the epigenetic state as a function of time in the same way that mutation rate functions with time. And it's another example of a biological process, which is an error-prone process, like every process is, with time. And that may be a reasonable hypothesis to start with. It may have nothing to do with cancer in that case, except that you're probably, just as genetics has to do with predicting cancer, certainly epigenetic changes may predict cancer, and so the accelerated rate would predict the cancer 15 years down the line or something like that. That would be, so these spots, the context of these spots may become more and more interesting in some way. That's could become an important variable here.
I mean, that makes sense that these are kind of hotspots for errors in terms of cost.
I had one other question, which I was really lying in wait to get the chronological age of an induced pluripotent stem cell, right? And the reason I say that is the following. There is an imperfection in the Yamanaka experiment.
I did have the induced pluripotent.
What is, I know you did.
It was 0.
Yes.
Yeah.
So let me tell you why I don't think it should have been 0. Okay. So there turns out to be an imperfection in the Yamanaka experiment. And the imperfection is that it doesn't wipe out. It isn't like meiosis. It doesn't wipe out all methylation. There is a residual methylation. And you can see that from, let's say, genetic predispositions that give rise to altered methylation patterns. They actually persist in some of these cases. So I would have thought that you might be a year old or you might be something plus. Well, 0 might be, 0. 0 is birth.
Yeah, it was
0.
It was setting it to birth.
I see. So yes, presumably if it had erased everything, you might have been minus 9 months, right?
Yeah, it was on part of the embryonic cell.
Minus 9 months is the time at which you're really naked epigenetically, right?
It actually was a little bit minus.
Oh, it was.
Yeah, I mean, it's gone. It was on par with the embryonic stem cell age.
It's interesting.
We have to better, we have to think we have to move on.